Thank you very much for this opportunity to participate to this very interesting workshop. So I am still in Institut Cochin in Paris, and Institut Bourgogne is the address of my collaborators, Olivier Piétrement. So in fact, we were interested in sepiolite maybe to be able to transfer DNA into mammalian cells. And because sepiolite, because of the structure of the sepiolite with channels and tunnels, there's a wide surface of interaction with different molecules, and it has been shown that it's able to interact with a lot of biological molecules such as sugar, polysaccharide, protein, lipids, and I will be interested in nucleic acids. And with a lot of potential biomedical applications. And this, but because of this fibrous structure, the people this raised concern about the potential toxicity or asbestos-like effect. And if you want to use it in biochemical application, we need to investigate a bit on this aspect. And but for the moment, I classified sepiolite as a non-toxic and non-carcinogenic. While for the biotechnology for gene therapy, there is a large number of possibilities for therapy of genetic disease, but also maybe virtually almost all kind of disease. And we need to have a vectorization to deliver the correcting genes into the cells. And there is several methods existing. Most can be divided into viral and non-viral method. And viral method, you can imagine there is some concern, some problem because you use viruses. But with non-viral method, we have chemical methods and physical methods. But one of the issues is that we need to have a balance between efficiency and toxicity, and doesn't work anymore. And maybe it could be interesting to have some kind of cargo that would be able to transfer at the same time the DNA and other kind of biological proteins or chemicals, for example, antibodies to target the cell. So that's why we believe that maybe using nanoparticle vectors might be interesting, and the reason why we think that sepiolite might be a promising vector. But to do that, the sepiolite need to penetrate into the cells without friction, no Yoshida effect like in bacteria, because the cell mammalian cell will not survive to such friction forces and will scratch, will explode. So the question is, does sepiolite, if we add just sepiolite into culture medium, does it penetrate into the cells? And is it possible to use it as a vector for nucleic acid result? And what would be the toxicity? And what would be the cell response to sepiolite interaction would be the topic of this talk. So the sepiolite we use comes from the close to Madrid, and then what we analyzed that the size, the length was the maximum of the most fibers. The length was 200 nanometers with, but where there is few fibers at 1 micrometer. And the width is around 15 nanometers. So, and then we got the opportunity, we used the opportunity that sepiolite contain a natural and spontaneous fluorescence to analyze and follow its behavior into the cells. And this is an experiment of video microscopy. So with a fluorescent microscope, and you can see the internalization of sepiolite into the cells. Okay. And another example here, you will see that here, if you see this molecule, these particles, they will be excluded, rejected from the cells, and then reincorporate into another cells. Meaning this is a very dynamic process. This is a very dynamic process, and that the sepiolite can spontaneously internalize into cells. They can be transferred from one cell to another, and then they also can be rejected from the cells. And that might explain why sepiolite is not so toxic, because it can be rejected from the cell and can get rid of the particle. In contrast with longer particle that can stay stuck into the cell and generate inflammation. Here is another picture where in gray you can see the cells here. In blue we stain the nucleus with DAPI, and in green you have the natural fluorescence of sepiolites. And you can see that sepiolite is mainly into the cytoplasm and never into the nucleus. That meaning that it might be interesting that to sepiolite into the nucleus, because into the nucleus it can affect and alter the genetic material. So here with transmission electron microscopy, you have the cell here, the nucleus here, and the cytoplasm around the cell nucleus. And when we add sepiolite, we can detect the fibers into the cytoplasm of the cells. And then what is interesting to notice that the fibers are embedded into membranes. This gives the idea how the sepiolite got into the cell. You can see here a fiber of sepiolite here, and the invagination of the cell membrane. Then we then embed the sepiolite and incorporate it into the cells. Here is one example of a sepiolite directly piercing the cells, but these events are very poorly, are not really frequent. And then another process, you can see the sepiolite fibers in black, and then you have pseudopods of the cell and cell membrane. Then these pseudopods can surround the particle, and then the particle is going to be incorporated into the cells and into the membranes, meaning that the main process for sepiolite to be integrated into the cells is what we call the endosome process, the endosome pathway, and then the sepiolite is present into endosomes into the cytoplasm. The point that if we want to use it as a vector, we need first to be able to bind DNA onto the sepiolite, and then to see whether it can go into the cell. So I will not detail all the experiments, because we have a complete characterization of the sepiolite and DNA interaction. This is some pictures by transmission electron microscopy. Here you have the fiber of sepiolite, and here you can see the molecules of DNA all around the sepiolite here and here. So to make a long story short, what we found that in fact DNA interact with sepiolite on the channels, not in the tunnels, through the silanol groups, and this need cation, and this intrusion can be displaced by adding chelation of cations such as DTA and so on. So you can bind the DNA on sepiolite, and then the DNA can be removed from the sepiolite. So what we tested to see whether we can transfer some genes into cells with the sepiolite, we used a plasmid DNA encoding for a gene giving resistance to the cells to an antibiotics, to G418. So when we put G418 in the cells, they die. If they have incorporated this plasmid DNA, they should resist. So So we assemble the plasmid onto sepiolite, and then we add this suspension to the cells. We add the selection, and then we let the for 2 weeks, 10.
J'ai très peu d'envie.
To have the colonies showing the cells that are resistant to the drug and that have incorporated the exogenous DNA. So when we had the on DNA alone, of course, we don't have anything, no resistance. Sepiolite alone, no resistance. And when we had the sepDNA biohybrid, then we can have different colonies, cell colonies, resistant to the drug, meaning that even we have a stable transfection of exogenous DNA 10 days after that are present into the cells and that confers the resistance to the drug. So what was most of these transfection, DNA transfection techniques are generally toxic to the cells, and here is the toxicity we measured with sepiolite after one day or 2 days, and then what we can say that the dose, the concentration of the suspension used for the transfection experiment, we do not have ever so much toxicity. At this concentration, the toxicity is very weak. So what are the cell response? What could be the cell response to the presence of an exogenous, a foreign particle into the cytoplasm? Well, when you have a stress to the cell, the cells need energy for all the pathway and the processes to manage this stress. And this energy is given by the ATP, adenosine triphosphate, that is made by the respiratory chain, and the byproduct of a respiratory chains is a reactive oxygen species. And reactive oxygen species can also make mutations in genomic instability. But the ROS are not always deleterious. They can be and all-core mutagenesis and genetic instability can lead to aging and cancer. But the ROS can also be used for the beneficial of the cells in different kind of mechanisms, and they can be used as secondary messenger to activate protective mechanism, and for example, in the case of the ROS, to activate the detoxification pathways. Another stress is the stress to the DNA damage that can alter directly the DNA, that this will lead to cell death. But if the DNA damage response, cells possess their capacity to respond to DNA damage and to repair DNA, but if this doesn't work, or if there is too much damages, and if the cell death is not working, then you will accumulate genetic instability that will lead to aging and cancer predisposition. So an exogenous treatment can also affect directly the DNA. And also when there is an exogenous foreign body into the cells, you can activate the innate immune system that will generate cytokines, modify the environment, and recruit the immune cells to eliminate the cells with this containing this exogenous molecule or particle like virus, bacteria, and maybe sometimes particles. And this is also the basis of immunotherapy. But if this is not working, of course, you will not eliminate this damaged cell, and then you will accumulate mutagenesis. That's the reason why the deficiency in the immune system is also frequently accompanied by cancer predisposition. But the problem is that if you have a chronic persistent of the stress, then you can accumulate inflammation, and the inflammation, if it's persistent or chronic, that also can lead to aging and cancer. So here we wanted to address the cell response presence of sepiolite with a measure of reactive oxygen species and the DNA damage response and the cytokine production by cells. For the ROS, we use a probe that we put the probe into the cells and become fluorescent when it is oxidized, and then you can measure by flow cytometer. You have a shift in the peak of fluorescence, showing the presence of ROS. With sepiolite, when 1 day or 2 days after adding sepiolite, we can detect the production of ROS at this concentration here, and at lower concentration at 2 days after the exposure to the sepiolite. So here we have the quantification of ROS production in this human cell line, which is osteosarcoma cells, and then we have the production of ROS 1 or 2 days after exposure to sepiolite as a function of the concentration, meaning that the cells detect the presence of sepiolite and they are able to respond to answer to this presence. Then what can be the response when we have a stress? Is this mutagenic or not, or is sepiolite able to generate DNA damage? When you have a break DNA damage into the DNA, exogenous or endogenous stress, then you have the DNA damage response, which coordinates cell cycle with DNA response, with DNA replication and DNA repair to maintain genome stability. In other words, the cells are arrested in their progression through the cell cycle, and then this gives time to the repair machinery to fix the problem on the DNA. Or if there is too much DNA, to engage senescence or cell death to avoid the proliferation of damaged cells. But if this doesn't work, then you have chromosome arrangement, mutagenesis, and cancer predisposition. So does sepiolite generate a cell cycle checkpoint defect and senescence and cell death? So the cell cycle, you have the S phase where DNA is replicated, and the mitosis where the chromosomes are segregating in the daughter cells. In between, you have the G1 phase that has the phase that prepare to S and mitosis. So when we measure the content into DNA, the DNA content, then you have the DNA content here in G1. Then you have replication of the during the S phase, so the content is increasing. In G2 phase, you have the content is double because you have replicate DNA, and then after mitosis, then you come back to the haploidy. So if there is the activation of the checkpoint, then you have the arrest in G1 or in G2 phase. But with sepiolite, the cells is able to detect the sepiolite, but there is no effect on the cell cycle distribution, suggesting that there is a lot of genotoxic stress. After its genotoxic stress, you have the activation of the cascade of kinase that will activate ATM or ATR that will phosphorylate CHEK2 or CHEK1 protein that then will lead to the cell cycle arrest, DNA repair, and so on, and also to apoptosis. So what we measure the phosphorylation for CHEK1 after treatment that we know that as a control activate phosphorylation of CHEK1 of CHEK2. Here is in transformed cells, cancer cells. Here is the primary fibroblast, normal cells. But with the sepiolite, we have no activation of CHIK2. This confirms that at least this pathway is not activated by sepiolite. There is no activation of the DNA damage response, probably because there is no DNA damage. Also, I will not show you, but we have some data that's showing that apoptosis is not activated. So this suggests that there is no or very few DNA damage induced by the sepiolite, which is consistent with the fact that we do not detect sepiolite into the nucleus. So, but the cells are able to detect the sepiolite because they produce ROS. Generally, cells also produce cytokine, and the cytokine will recruit cells from the immune systems. So the question is, are now the cells able to produce cytokines when we put a sepiolite on this? So we measure the expression of genes coding for different canonical cytokines after the presence of sepiolite. And then we can see with two different concentration and either 1 or 2 days after exposure that sepiolite induce the expression of genes coding for inflammatory cytokines. So to summarize this, sepiolite can spontaneously goes into the cell through endoplasmic system. The sepiolite is recognized by the cells, but and produce ROSs, and also activate cytokine genes that produce cytokines that might be able to modify the microenvironment of the cell. That reactive oxygen species are also able to make the cells produce cytokines. And this should stimulate the innate immunity. But we do not have any evidence that there is direct DNA damage because there is no DNA damage response. So this suggests that there is no or very low level of DNA damages. And because sepiolite can be rejected by the cell, this response should be very transient. And maybe we can expect to use this to exploit to increase the efficiency of immunotherapy. We can also speculate that with longer fibers, the fibers might be stay stuck into the cells and not rejected and is not transient, and then we can have a chronic production of inflammatory cytokine and inflammation. And that question that reactive oxygen species might be able to do to generate genetic instability with a chronic presence of this molecule. And then that maybe generate cancer. So the question is that in the first preliminary experiment to see whether we are able to do something with immunotherapy stimulation. Oh, no, first, we have seen look at the production of cytokine by different kind of clays, mineral. Here the result with different cytokine here. The induction of cytokine with sepiolite, and with this is the lapionite with halosite, nucleoside. So the point that there is all the clays we have tested are able to generate the induction of inflammatory cytokine genes. Well, it's depend on the concentration, the time of contact, and so on. But we have the more efficient are sepiolite and chlorisite. And if we compare to nanodiamonds, we can see that in fact nanodiamonds are also they produce inflammatory cytokine, but more or less efficiently than the clay minerals. Then in a preliminary experiment. This is a preliminary experiment because we used for this experiment very few mice, only 6 per point, and because we need to follow the regulation, the rules for the experiment with animals, and to see whether it can be interesting to make more deep analysis. But we have injected mouse cancer cells, MET1 cells, into mice, and then we obtain this cancer. This is immunocompetent mouse because the cancer cells comes from this same mouse strain. And then after 1 month, we got the tumors, and then we injected sepiolite with the antibody against PD-1, which is an inhibitor of the immune checkpoint. This is the basis of immunotherapy. The tumor should be sensitive to this antibody. And then what we observed that in absence of sepiolite, the tumors grow because these tumor cells are known to be cold to immunotherapy. And then they are not affected by the presence of PD-1. But in the presence of sepiolite, we do not have regression of the tumor, but the progression is stopped. If we looked at after 22 days, this is this point, the growth of the tumor without sepiolite, with the antibody, the tumor grow. With sepiolite alone, without the antibody, there is no effect. But when we associate both of them, we have an arrest of the tumor progression. This has to be repeated with more mice, maybe to try different concentration of sepiolite, different conditions. And what we expect at 1 day is to have and to detect and to have tumor regression. That would be the goal, and we have to verify that when we have the invasion with cells from the immune system. But this is a very encouraging preliminary experiment. So this work was done by Elodie Dardiac and Sandrine Ragu in my lab at Institut Cochin, former Gustave Roussy, with a collaboration with visiting scientists from Cuba, Fidel Antonio Castro Smirnov and David Adam Bruce, and with a very nice collaboration with Eduardo Ruiz-Izqui and Pilar Aranda that opened me to the fascinating world of the clay minerals. We also have this collaboration with Eric Lecan and Olivier Piétrement for the transmission electron microscopy, and Jean-Rémy Bertrand for the nanodiamonds. Thank you very much for your attention.
Thank you very much, Bernard. Nice work. I don't know if there are questions. We have not too much time, but can have some questions.
I have one, and actually it is the same question. Thanks a lot for this excellent presentation, but what concerns me is why your sepiolite is fluorescent. I think I already asked you this question during ECS meeting, and after that I was fascinated by the fact that it is intrinsically fluorescent. So we checked with all microscopy means available in our lab if our sepiolite is fluorescent, and it wasn't. So what is so special with your samples you were working with that they appear to be autofluorescent?
Yeah, I don't know. It's that sample that come from Tulsa, that Panjal S9 given by Eduardo and Pilar, and we haven't made any we didn't do any modification, functionalization. It was just we opened up. Though maybe it depends on the wavelength of excitation and emission. I don't remember exactly the wavelengths. They are in the paper we published with in a scientific report. And But the sample we got from this sepiolite from close to Madrid is spontaneously fluorescent, and we didn't do the exact wavelength. I can, if you want to send me an email and give you
I will email you
the exact value, because I don't have them in mind, but I can find them to send you these values.
Thank you.
Actually, Olivier also talked in the chat. We don't know why. This is a sepiolite from Madrid. Maybe they have some elements, residual element that had the fluorescence, but we found this, at least in our work, was because we were trying to put to fluorescence, and we observed always like a background when we used the sepiolite. So it was, for us, was bad, but it looks like for Bernard is good.
Sigma sepiolite is not fluorescent at all at any wavelength, so I suggest you
because
use
it's
Sigma
not. It's
1.
probably from Turkey, not from Spain.